Eukaryotic cells differ from prokaryotic cells in that they contain a nucleus and other membrane-bound structures called organelles. Eu- the endoplasmic reticulum is a contiguous complex network of interconnected tubes and membrane sacs that extend through the cell's cytoplasm. This complex makes up roughly 1/2 of the membranous tissue of the cell. There are two types of endoplasmic reticulum, the rough and the smooth. The rough endoplasmic reticulum, or rough ER, gets its name from the fact that its surfaces are studded with ribosomes. The membrane of the rough ER is contiguous. The nucleus is a membrane-bound organelle that contains most of the cell's genetic material. The interior of the nucleus does not contain any membrane-bound organelles or compartments. Its contents are non-uniform and consists of a number of subnuclear bodies like the nucleolus. The genetic material in the nucleus is organized as multiple linear DNA molecules combined with a variety of proteins to form chromosomes. The gene Nuclear pores are hollow protein structures that penetrate the nuclear membrane and act as doorways between the nucleus and the cytoplasm. They allow small molecules and ions to freely pass into or out of the nucleus, but also may allow larger proteins to enter the nucleus from the cytoplasm if the proteins have special sequences, or tags, that indicate they belong in the nucleus. In the same way as mitochondria are the power plants of the cell. They convert sugar into ATP and enzymes related to cell metabolism. Mitochondria are unusual in that they are surrounded by two membranes and contain their own independent genome. They also can divide independently of the cell in which they reside, meaning that mitochondrial replication is not tied to cell division. The number of mitochondria found in a cell varies by the type of cell. The more energy a cell needs, the more mitochondria. The cytoplasm is a semi-fluid, gelatin-like substance that surrounds the organelles inside the cell. It acts to cushion, protect, and support the organelles, and fills all the empty space with the cell membrane not occupied by organelles or other structures. Although the cytoplasm is made up mostly of water, its structure and properties are not fully understood. There are multiple levels of Inside each human cell, there is approximately 6 feet of DNA existing as 46 chromatin strands. Normally, this DNA is dispersed in a somewhat loose configuration. However, when the cell is preparing to divide, each strand of DNA is condensed by a factor of 10,000 times into a structure called a chromosome. The nuclear membrane consists of two lipid bilayers, the inner nuclear membrane and the outer nuclear membrane. The function of the nuclear membrane is to separate the nucleus and the nucleolus from the rest of the cell contents. The nuclear membrane is embedded with special proteins called nuclear pores, which act as specific doorways that permit substances such as nutrients, waste, and cellular information to pass into. The centrioles are a pair of structures on either side of the nucleus made up of 9 triplets of microtubules. The centrioles play a key role in the formation of the mitotic spindle during cell division, as well as helping determine the spatial arrangement and location of the nucleus. The lysosomes are tiny sac-like structures filled with enzymes called hydrolases. Like vesicles, lysosomes are constructed of a phospholipid bilayer similar to the cell membrane. The The lysosomes function as the digestive and waste disposal organelles of the cell and enable the cell to utilize nutrients. Lysos The endoplasmic reticulum is a contiguous complex network of interconnected tubes and membrane sacs that extend through the cell's cytoplasm. This complex makes up roughly 1/2 of the membranous tissue of the cell. There are two types of endoplasmic reticulum, the rough and the smooth. The smooth endoplasmic reticulum, or smooth ER, gets its name from the fact that it lacks the surface ribosome. The Golgi apparatus consists of stacks of folded membranes called cisternae. Each disc-like cisternae contains different enzymes which selectively modify the macromolecules that are passing through them. The Golgi apparatus The cytoskeleton is made up of a series of specialized proteins called microtubules and is the cellular scaffolding or skeleton contained within a cell's cytoplasm. Much like our skeleton, the cytoskeleton provides structure and facilitates movement of the cell. It also plays important roles in the cell membrane physically separates the interior of all cells from the outside environment. It's made up from a phospholipid biolayer embedded with specialized proteins. These specialized proteins act as doorways into and out of the cell. The cell membrane is selectively permeable to ions and organic molecules. The ribosomes are large, complex molecular machines found within all living cells. Their primary function is to synthesize new proteins by reading the instructions from a messenger RNA strand. Ribosomes do this by linking together amino acids in the specified sequence. Ribosomes consist of two subunits, each made up of ribonucleic acid, or the nucleolus is a dense, spherical, non-membrane-bound structure found in the nucleus of the cell. It's composed of primarily proteins and nucleic acids. Its main function is to trans-5 ribosomal RNA and combine it with proteins to form ribosomes. The nucleolus Vesicles are basically mobile storage units. They are similar to lysosomes in that they are constructed of a phospholipid bilayer, but differ in that while the lysosomes are involved in digestion and transport, the vesicles perform no modification.